we'll do is we'll get started with Regan, Baird. Dr. Baird actually received his PhD from Temple University in Philadelphia in biochemistry. He did a postdoctoral fellowship in the Beth Israel Deaconess Medical Center in Boston, Massachusetts. Dr. Baird has spent the past 2 decades in cellular and tissue imaging and analysis. In fact, I think it was about 2 decades ago I met him because we had a spinning disc system while he was with a company called 3i. So right now he's currently Visiopharm Sales Director of North America East, and he's an excellent resource on the practical aspects of artificial imaging, machine learning, deep learning, tissue multiplexing, and clinical integration. And I had an opportunity to try their Visiopharm's multiplexing software on plant materials, and I thought it would be really helpful for folks to understand, what's out there commercially. So go ahead, Regan, and we look forward to hearing your talk.
Oh, thanks, Kirk. Hopefully you can hear me. Thank you for the invitation to speak. Hopefully I'm coming in loud and clear. Let me get the screen going. I wish this was. So as you mentioned, I'm going to be talking today about multiplex analysis workflows, because we can, as you've seen, generate lots and lots of really cool images using lots and lots of cool techniques. But how do you get data out of all of these images, and how do you approach analyzing images that are full of data? And I'm gonna, like most of my colleagues, focus a little bit more in the animal space, but hopefully I can relate a little bit of this to plants, thanks to Kirk. And I might show some of the images. I don't know if I have permission to show, Kirk, but they're in this presentation. And the software platform that I want to talk to you guys is something called Oncotopix Discovery. Don't let the word onco scare you in thinking that this is a cancer-related software. We at Visiopharm, we focus on cancer biology, and creating AI to look to detect cancer and cancer-related projects. However, the tools that we use are universal in being able to analyze tissues of all shapes, sizes, and species. And the technology that we really want to leverage in order to make image analysis and getting data out of these digital images very easy is a technique called deep learning. I'm sure a lot of you have heard about AI and deep learning in the field. And what we're trying to do at Visiopharm is democratizing this technology, so anybody that has domain expertise in their tissue can use deep learning, because if you can see the objects in your image, you could train the computer to find them. And once the computer's found them, you can do a lot of quantitation and to be able to convert your images into graphs, because one image is pretty, but many images make a graph. As we have more and more experience with this deep learning technology, we are starting to create some pre-trained algorithms that can be used. Now, as I said earlier, we're focusing on cancer biology, so a lot of our pre-trained algorithms focus on that. However, you can literally train the software to find any of the different types of structures, whether they be macro structures in the tissues that you're looking at, or cellular and subcellular structures. And the software is actually quite easy. If you can see the structures that you're looking for, here I'm showing a fluorescent image, but this would work in chromogenic images or any type of the different imaging modalities that you've learned about today. You simply have to provide the software with annotations. In this case, we have 2 tissue compartments that we're interested in segmenting. And we simply paint over those objects. We give the software many examples of the different areas that we'd like it to find, or the different structures, different objects that we'd like it to find. We push the train button, and iteratively, it will learn from that training set what the different objects are, so that when you provide it examples that it hasn't seen before, it will be able to demarcate those specific objects and find them in places it hasn't seen before. The common question is, how many do I need to provide it so it will learn? And the answer is not easy. It really does depend on the morphological and intensity variation in the different types of objects you're looking for. We can talk more about that if you have specific questions at the end. And in order to do multiplex analysis, at Visiopharm, we really look at a multiplex as basically several individual stains, several rounds of individual images that are all stacked on top of each other. So everything that we have learned in our domain expertise as cancer biologists, where we're looking primarily at what we call immunohistochemistry images, in a multiplex world, we're just looking at many, many, many of those stacked together. And it doesn't matter if they are staining cells, staining compartments, staining subcellular structures. We see each of those as an individual channel in the image, and so we can analyze them. And the workflow that we've developed with our software is really to leverage the power of this deep learning technology. So in the first step, you're going to do a tissue compartmentalization. And I do apologize that I'm going to have a cancer-specific example in this presentation. However, I think you can extrapolate to other fields how all we want to do is, if we have a big picture of tissue, we want to segment it into different areas. There are ways of doing this based on contrast. There are ways of doing this based on morphology, and there are ways of doing this on the spatial relationship of those different compartments that I won't get into today. Now, the second thing that we're going to want to do is we're going to want to segment the objects that we're going to use for the biomarker multiplex analysis. And we're going to want to do that because we want to understand the stain within each object. In this particular example, I'm going to talk about my objects as cells. In animal biology, obviously, cells have a nucleus, a cytoplasm, and a membrane. And so in this particular case, we're going to be able to segment cells such that we can outline them based on their cellular structure and cellular morphologies. This actually becomes the most important step in any downstream phenotyping analysis that we want to do, because anything that's contained within the boundaries of that object or that cell is any of the signals contained within those boundaries are going to be assumed to be a part of that cell. And so we really need to understand how to do the best segmentation of each object or each cell in this particular example. In fact, this can be the most challenging part of all of image analysis in tissue today. I'll show you a little bit more about how we can approach this with the Visiopharm software. The next step we're going to do is take that segmented image. And actually, at this point, if you wanted to, you could simply say, for each segmented object, please give me the expression of every single biomarker or other signal within the boundaries of every single object, every single cell, and give me its XY location. I can generate a massive data table for the billions of cells that I've identified. I can hand that off to my data scientists or whomever else, and they can work with data tables to give us some quantitation. Alternatively, within our software, we can go through our multiplex analysis, where we have a couple of ways where we can have the software assign phenotypes based on the expression patterns of individual objects so that we can reclassify them as individuals and then do a bunch of quantitative analysis where we're going to generate simple results. Maybe we want counts, percentages, some things like that. I'm going to show you a way that we can do some very cool data visualizations so we can project heat maps so we can look for areas of high density. Again, we know the XY coordinates of every single object, so we can do some cool object distance calculations and neighborhood analyses. And in Visiopharm, we have now developed some very cool interactive bidirectional plots so that if I click on a point in a graph, I can see those objects in the image and vice versa. And this really gives me ways of being able to do some amazing quality control over the tools that we're using, and to give me a way of exploring data that isn't found in most pieces of software. So rather than give you a sales pitch and talk about, all of the things that we can do, I'd like to give you some real world data and talk about a true experiment. In this particular case, we have an image from a device that you're actually going to learn about in the talk after me called the Codex, where we looked at 20 individual protein biomarkers plus a cell specific nuclear stain called DAPI. And we're going to go through the process of how you would phenotype billions of cells using as automated a process as possible. And one of the very first steps that we want to do with any piece of tissue, and who cares what this tissue actually is, it happens to be cancer. However, that's not important. We want to do some algorithms to check the quality of the image. And we might want to identify areas that are blurry, areas that might be rips, tears, and fold in this tissue. And here you can see we have an area that we definitely think is an artifact that we want to have the AI identify and remove from further analysis. And so what we're going to do in Visiopharm in Oncotopix discovery is create an analysis pipeline. And in that first stage of the pipeline, we want to find any artifacts. The way this is done is by grabbing many, many, many images that might have the different types of artifacts we're looking for, and we're going to train the computer to learn what are these artifacts and what is good tissue. That may be different between looking at cancer patients versus looking at plants of different species and different imaging modalities. But basically what we would like to do is have a pre-analysis check of tissue quality and eliminate areas that shouldn't be analyzed or are questionable for analysis. Remember, the ultimate goal is to create an analysis pipeline that we can run all of our images through so that we can generate massive amounts of cellular data instantaneously. So we will train the computer to find these artifacts and then run that algorithm over all of our images. The second thing that we're going to want to do in the second stage of our analysis pipeline is to have the software find the different macro compartments that we're looking for. This is an immuno-oncology example. We're looking for immune cells that are penetrating the tumor, that are near the tumor, and that are outside of the tumor. And so what we want to do is have the AI automatically compartmentalize our tissue by training it on what is the tumor compartment and what is the non-tumor compartment. If you're in a different field, obviously you can extrapolate to what compartments you might be interested in training the software to find. In the case of the margin, we ask the software, hey, once you've found that tumor area, please expand a set distance around that tumor and give me another area that is not specifically stained for, and let's call that the margin, or the area between the tumor and the non-tumor areas. And the reason why we've done this is because we want to quantify all three of these as independent areas, so independent calculations. And you can have a lot of heterogeneity in the tissues that you're looking at, and so you need to have an AI that can automatically do this demarcation for you. So we've trained it on several examples and validated that it works across the different areas of interest. We're then going to set those as what we call regions of interest for independent analysis. So anything that's in the yellow will be analyzed as one thing, anything in the pink and anything in the green would be analyzed as different things. Once we've done our compartmentalization, we now want to identify the cellular objects within the tissue. Again, you don't necessarily need to identify cells. You could identify any type of organism or whatever object you're looking for. Obviously, in cancer biology, we're interested in cells, and we want to draw a line around every single cell. These are fairly small objects, and we want to create an algorithm that's going to do this. In the case of animal biology, had a close-up, maybe it's here, there it is. Sorry, I'm going back. I made a mistake in my presentation, but here's a close-up of our ability to do this segmentation. And what we have done is we have said, okay, all of my cells have a nuclei. Our nuclei is stained by DAPI. So let's create an artificial intelligence algorithm that knows what DAPI stained cells are, find their nuclei, and then expand that nuclear signal so that it goes to the edge of the cells. That expansion can happen a variety of ways, one of which is just saying, hey, go a certain distance. Other ways that that can happen is by saying, I know that I have these other signals in my cells, so please expand by those signals until you hit the edge of the cell. There's a lot of cool ways that you can do this type of segmentation. When you get down to this resolution, it can be very challenging to get perfect segmentation, but I think you can see that this algorithm has done a fairly good job of finding the individual cells in this field that we're looking at. It could also be improved. And if you find that the algorithm that we have designed for the nuclei detection doesn't work for your specific sample, you can always retrain that algorithm to find all of the different cells in your sample. I'm going to go a little bit. Now, what if you don't have a nuclear signal, or you don't have nuclei that's stained by DAPI like we do in the mammalian world? Well, we can actually develop algorithms that we want to perform, to give the computer the rules that we want it to perform on. And so here's an example that Dr. Czymmek gave me, where he sent us a multiplex image. I don't know anything about it other than it looks beautiful. And what we did in the Visiopharm platform was take all of those individual signals and combine them, the appropriate ones, into a single channel that we could use as our cell wall signal. This image actually reminds me a lot of human muscle tissue. And so we used some of the same tricks that we understand from human muscle tissue to be able to identify the individual cells. And so you can see in green here, we have identified all of the individual cells in this plant tissue. So you don't necessarily need to work with the DAPI specific signal as I had shown in this, the tumor example. Once we have put a line, a border around all of the objects that we would like to understand their phenotypes, and by phenotypes in this specific case, I'm going to talk about what biomarkers they're expressing. You could take that a step further and say not only what biomarkers are they expressing, but also what genetic signals are they expressing, because I might have those signals. You could take it a step further and say what metabolites are they expressing, or are they high and low in? Whatever it is that you're interested in including in your phenotypic description. The first pass is we're going to identify the different phenotypes based on the presence or absence of signals. Again, I don't care how those signals are generated because this is an image at this point, a digital image. We just know that each signal has a high, medium, and low intensity range. You could have more degrees of that. But essentially, I'm going to say, I'm going to set a level of positivity by an intensity threshold. And what I can do is just simply say, if a cell is above or below a certain signal, for signal A, call it positive. For signal B, call it positive and change its color. For signal B plus C, call it positive and change its color. And I can do this manually and create the phenotypic tree that I am specifically looking for. This is quite easy if you only have a couple of signals. This becomes arduous if you have a lot of signals. And if you have a lot of signals, you might not want to do this manually. You may like to push the button that says auto phenotype. And when you press the auto phenotype button, the software will tell you in return, hey, these are all of the different phenotypic classes that I have identified by a technique called auto clustering. We're not going to get too much into what auto clustering is. I suspect the speaker after me may speak a little bit about this as well. This auto clustering will then provide phenotypes that it has found. You will get a list of those phenotypes, and in that list, you can then go back and troubleshoot to say, hey, these don't make sense. Let's merge these. Let's combine these. I can also take this information and say, I'd like to incorporate morphological, intensity, or spatial information into the phenotype. So, hey, if I'm a cell that's positive for X, Y, and Z, and I'm also a certain size, convert me to a new phenotype. If I contain X, Y, and Z, and I have a high level of A and B, give me a new phenotype. If I am an immune cell within 20 microns of a tumor cell, then give me a new phenotype. So you have the ability to start to add to the phenotyping information rather than just being positive or negative for different signals. Once you have your phenotypic classes defined and identified, now we can start quantitating, and you can quantitate within each one of those compartments as we had. It's very simple to set up the quantitation. You just ask the software to tell you the values for certain different types of counts or signal or whatever it is that you're looking for. You just query for those different metrics. You create the data table that you're looking for, and when you press the batch process button, it'll run through the analysis pipeline and give you all of these results. This is just showing that you can plot those results. This was in Excel, so you have all of the data is extractable from our software. It's not locked onto our platform. Once you've identified all of those phenotypes, you may like to do some additional visualizations, like, hey, where is the highest density of phenotype A? And is that interesting in my image? And you can create a very beautiful visualization to tell you exactly where the business end of that specific cell type is located in your greater tissue. You can do this for multiple different cell types. You could even combine phenotypes to say, where are these 2 or 3 different phenotypes interacting together? This is an old plotting system. We're not going to look at that. The other thing that we can do is we can start looking at some data reduction plots. And I'm not sure if anybody else has presented t-SNE plots or UMaps before me, but basically what we can do is we can take all of that multi-dimensional information and squash it down into a 2-dimensional plot. The graph on the left has nothing to do with the shape or the location of the objects on the right. They're not, but what we have done is we have color-coded every single point in the graph is representative of a cell in the image. And this starts showing me the clusters that have formed due to my auto clustering analysis, even my manual phenotyping analysis of how these different phenotypes cluster in relation to each other. If you've ever done this in flow cytometry, you get beautiful separation of clusters. In tissue, we're not always that lucky. But what's great about the way our software handles this is I can gate on a population of cells in the t-SNE plot, and it will actually show me those cells and put a box around every single cell in the image that is within that gate. So I have a bi-directional relationship between the plot and the image. I can click on a cell in the image and it will show me where that data point is in the plot. And so I really have a way of understanding what's happening in the single image, or if I run a t-SNE plot on multiple images, I can have a bi-directional experience with all the images in my data set. I can actually take this a step further and start filtering information from the 2-dimensional reduction plots by looking at the individual compartments, and do I start to see differences between these different compartments? If I have different cohorts of treated and untreated samples, I can separate between those. I can look between different images, and I can really start to explore this data in ways that if I just look at images, it's hard to understand because you have to remember from image to image what you were looking at. These plots and the way that you can interact with these plots to go back and look at the individual objects in the image gives you a very dynamic and interactive way to troubleshoot and explore your data deeper and further. When it comes to the spatial relationship, remember I told you we know the XY location of every single cell in the image. I can also plot the distance of every cell to a specific cell type. So in this particular case, we're looking at the distance of immune cells to the tumor. In immuno-oncology, there's a relationship to the disease and how you would treat different patients depending on the infiltration of their immune cells. And I can see exactly the distribution of every cell and its distance to a specific other cell type. And you can start to see how these different types of phenotypes are interacting with each other. I could actually take this a step further and say, okay, probably since I'm looking at tumor cells and the distance of immune cells to tumor cells, the cells at the bottom are the ones within that tumor gate, and I can remove the margin and stroma gates. And actually that's what you would see. I don't have those examples to show you. The intermediate cells are the ones in that margin region, and the ones that are the far outliers are probably the ones that are in that stromal area. Again, I can select any of the cells in these and say, hey, where's this pink outlier? Click on it, and it will show me that cell in the image that it came from, and I can verify that that cell is correctly phenotyped and is in the proximity that it's saying in the specific plot. To take the example that Kirk gave me a little bit further, we've done that segmentation based on cell wall. We have several signals built into this multiplex image. We can push the auto phenotype button and it will come back and it will tell me all of the phenotypes that are present for each one of these individual cell types that we identified using our segmentation routine. Very quick, very easily. And because I have all of these phenotyped, I can start to ask many, many more questions. And I can go back and check with the bidirectional plotting tools if I'm correct. So just to summarize the presentation, Visiopharm, through our Oncotopix discovery platform, has been able to leverage the power of AI and deep learning to go in and find and identify the objects and the compartments within tissues with very, very high fidelity. None of the data is locked within the software, so you can extract and take out as much as you need. But we have built in all of these additional visualization, plotting, and phenotyping tools to help you understand the high-plex data that you can create from all of these imaging techniques that you heard about and you'll probably hear about in the next presentation. I wanted to, again, thank you, Kirk, for the invitation to speak today. It's nice seeing you again. Thank you for the images for the plant biology. It's one of the only times I've had a chance at working with that kind of data. And I wanted to thank the audience. If there's any questions, I'm happy to address them now, or you can always email me through the organizers.
Thanks, Regan. We have one question at the moment. Are the customizable training algorithms commercially available, or are the user have to create their own depending on the research task? So I know you mentioned this a little bit, but maybe you could expand on that.
Sure, you have both options. We have a library of algorithms that we have developed. These range the gamut. I would say 99.9% are not in the plant field. Probably 100% are not. But anything that we have of content that we have created is available to the customers that purchase our platform. Those algorithms are all open and you can modify them to fit your specific needs if they aren't perfect for your research. And if those algorithms aren't giving you the precision or don't address the things that you have, it's an open platform, so you can also create from scratch novel algorithms. So you actually have all three options at your disposal. Okay,
and just to add a clarification point, if I had your system and I was working with some of my samples and I created one that was helpful for the plant community. I know you helped us when you were initially doing this. You could validate it and make it available for others. How does that get shared? Do you see what I'm saying?
I do see what you're saying. We don't make available our customers' algorithms to the general community. We just do the ones that we validate. However, they're small, simple packets, and you could email them to anybody that has Visiopharm, and they would be able to run those algorithms. It's not a challenging process to share.
Okay, excellent. Okay, Regan, thank you so much.